Okay. Welcome to this first summer school webinar that is organized in collaboration with EURO-NMD. As you know, the webinars are usually a production of EURO-NMD with the Rare Neurological Disorders Network and the European Academy of Neurology. This webinar will cover the features of clinical trials, in particular the ones for rare diseases, and the John Walton Muscular Dystrophy Center and Dr. Guglieri have vast experience in running those trials and coordinating a team of healthcare providers, project managers, and researchers with diverse skills to deliver the results that the patients require to improve. Michela Guglieri is a senior clinical lecturer and consultant neurologist at the Newcastle University and Newcastle Hospital's NHS Foundation Trust. She has a special interest in care management and translational research in inherited neuromuscular disorders and has been involved in several clinical trials in Duchenne muscular dystrophy and other neuromuscular disorders. And currently she's a study chair for two large international trials on DMD. I would like to thank Michela for accepting to do this webinar, and thank you very much. And hopefully everybody will learn a lot from you. I know you have a lot to teach. Thank you very much, and the floor is yours.
Thank you, Antonio, for the nice introduction and for giving me the opportunity to do this first webinar. As you probably know, we've done a similar session in the past, but obviously we're face-to-face. So this is the first time that I deliver this specific lecture online, which makes everything a little bit less interactive and therefore more difficult from my point of view. But I really hope that we can still benefit from it, and you will find the presentation interesting. So I try to share my screen.
It's on.
Okay, great. So as I mentioned, as Antonio mentioned, I'm Dr. Michela Guglieri, work in Newcastle in the UK, although clearly from my accent, I'm not originally from here. And my specific interest is really in translational research. And over the last 15 years that I've been working here, I've been involved in several clinical trials. What I would like to do today is really to, at the same time, really introduce in a way the topic of clinical trials. What is a clinical trial as a first message, but why do we need a clinical trial? A little bit discussion about the different phases of clinical trial, either if any of you would be interested one day or involved in one day in the development of a clinical trial or a clinical study, or whether you might be exposed in the future in terms of being involved in delivering a clinical trial, in consideration that will allow you hopefully to critically review clinical trial design and endpoints. We also spend a few slides talking about ethical concern and what is important to be addressed in the informed consent. So what are the messages? And I don't want to replace other webinar on and training courses on good clinical practice. I think based on my experience, what are some key points that should always be mentioned in the informed consent form and should always be discussed as part of recruitment in clinical trial. As I mentioned, I used to do this lecture face-to-face, and it was nice to have some discussion about case studies, which might be difficult in a group, although if we have time, I'm more than happy to do so. But what I thought is to make it a little bit more up to date and discuss about why clinical trial fail. My specific area of work is neuromuscular, rare genetic neuromuscular diseases. And specifically, I have a specific interest for Duchenne muscular dystrophy. And over the last few years, we've seen several clinical trials failing in Duchenne muscular dystrophy. And I really want to share with you this experience to learn and what can be done better, either in Duchenne or other neuromuscular diseases, but more generally about issues that might need to be considered when design or delivering a clinical trial. So first of all, what does it mean, a clinical trial? This is the definition from the World Health Organization, and means that a clinical trial is a research study that prospectively assign participants, a human participants, so it's done on people or on group of people, one or more intervention to evaluate the effect of this intervention on an health outcome or one or more health outcome. And I think that the key point is that in order to be considered a clinical trial, a study has to be prospective, has to be done in humans, and has to evaluate the effect of an intervention. But this intervention can be different. We are still talking about clinical trial, whether the intervention is a new drug, which is probably easy for all of us to understand, but also if it is a new device, a new method of screening, or a new diagnostic method. All of these can still be considered intervention as soon as they have an impact on a health outcome. Important to differentiate between clinical trial and observational study. An observational study is when, in a way, there is no external influence of what we do. So as an investigator is not changing the practice, but is observing the natural relationship between factors and outcome. That can be quantitative, can be qualitative, can be retrospective, can be prospective, but the important thing is that you observe, you record, you analyze, but you do not change or influence what you assess. So how can we really understand if you are in front? Because sometimes it seems to be very easy, but sometimes it's not so easy to understand. Should this be considered an observational study or a clinical trial? And the importance is that if you are the one designing an observational study or a clinical trial, they follow different regulation and different requirements. So it's important to understand, am I doing a clinical trial or is this an observational study? And then the regulatory process and the documents that are required might be completely different. So in order to define is a clinical trial, first of all, are we involving human participants? We are not talking about clinical trial that involve samples and surely not animal models. The other point is whether the participant is a study's prospective. So are you collecting data prospectively based on a specific outcome a specific design that you have developed? And have you assigned a specific intervention, whatever this intervention is? Are you really evaluating the effect of this intervention of the participant? And what are you measuring is in health-related biomedical or behavioral outcome. Can still be a questionnaire, can still be a quality of life outcome, but has to be something that is related to health. But one of the questions that I've been asked several times is, do we really need clinical trial? Because clinical trials are complex and, as you will see throughout my presentation, are costly and time consuming. And sometimes one of the questions is, we see this patient in clinic, we already do the intervention. Why do we need clinical trial and structured way to measure a drug effect? And I think the example for that perspective is really given by the use of corticosteroid in Duchenne muscular dystrophy. Corticosteroid have been prescribed in Duchenne muscular dystrophy since the late 80s and quite consistently since the 2000s. And are part of the care recommendation in 2010 and confirmed in 2018. So are part of the care and is now recommended that every child diagnosed with Duchenne should be offered the possibility to take a corticosteroid. Is the only treatment that is currently available that has been shown in short clinical trial of 6 months long, their benefit in terms of muscle strength and in terms of different aspects of the disease. However, though the benefit of corticosteroids has been documented, There are also, as probably all of us know, corticosteroids are associated with several side effects. And this has led to a great inconsistency in how corticosteroids are prescribed in Duchenne muscular dystrophy across the world. And this is just an example of different corticosteroids, the Deflazacort or prednisone, and different regimes. These are probably the three most commonly prescribed. But if you look into inquire different clinics, you can realize that there are much more regimes that are prescribed. And I would love to say that are prescribed based on the specific patient, so it's an individualized approach, but the reality is that different centers use different corticosteroid regime because of the experience of the PI more than evidences. So even if we've been prescribing corticosteroid for 40 years in Duchenne muscular dystrophy, we still have several questions. And first of all, which corticosteroid, prednisone or deflazacort? Which regime? When should we start corticosteroid? When should we stop corticosteroid? And this lead to the fact that in general, in Duchenne, patients are treated with different steroid, different regime, different time point. Some of them are continuing for their duration of their life, and in other cases are discontinued. And this creates the fact that some patients still after 40 years when a drug is used, some patients are treated suboptimally, so with low doses or not treated at all, and other are exposed to higher doses and therefore higher risk of side effects without having a rationale behind this decision. Moreover, if you do not know exactly, so if there is inconsistency in the a treatment that is part of the care recommendation, examining the effect of any other drug will be impacted by the difference in the corticosteroid regime. And even 40 years later, we are at the point now where we have actually drugs that are developed in order to replace corticosteroid, because in order to maintain the efficacy of corticosteroid, but reduce the side effects profile. And it took 40 years to develop something. And we have the first announcement just in June that there is a drug called vamorolone, which show initial positive results. But it's very difficult to develop a drug that wants to substitute an approved treatment when the approved treatment is prescribed totally inconsistently. What do we compare this with? What do we want to obtain? What are the real side effects profile of a drug? And how can we evaluate whether a drug is better or worse if we have so many questions on the original treatment? And the other point again from this is whether instead of conducting a complex clinical trial, can we just actually collect data from our clinic and our experience, our patient that we see? We perform assessment on a regular basis according with the standard of care. Why don't we just collect this data and reach the conclusion about what is the best treatment based on real life data? And I think that the field is changing, and I cannot underestimate the importance of real life data. However, when we look into conditions like rare diseases, when we need to involve a large group of patients from different countries, from different clinics, there are several limitations in the collection of longitudinal data in real life. A lot of data are missing. There is inconsistency or discrepancy between how people test the safety and efficacy of drug and how to monitor side effects and how to manage side effects. And therefore, the conclusion will always give a trend, but not a clear answer. And we conducted a clinical study, that is the 4DMD study, that is try to compare in a clinical trial of 3 years long, the 3 corticosteroid regime. But it was done so late, it took very, very long time in order to complete the study, and the results are not available yet, but hopefully published soon. But what I wanted to say here is that if the clinical trial would have been designed at the beginning of 40 years ago in a consistent way, we would have now several answers that we are still waiting for. Going more in a more didactic way, discussion about what are clinical trials. So we said that it is a study that is prospective and want to evaluate an intervention in human participants. But clinical trial are part of the drug development. So it's how a drug, or translational research, how a drug is taken from the lab to clinic. So how do I develop the drug? How do I test the safety and efficacy of the drug to justify that this drug is prescribed in routine in clinic as part of the care of patient? And these apply to any disease, and apply, although there are some differences for rare diseases, the structure and the process is identical, whether it's a rare disease or a common disease, whether it's pediatric or adult. So the drug development happens in phases, and obviously the first phase is the preclinical. So we need to demonstrate that the drug works in vitro studies that can either be in cells model or in animal models. But when we move into, we demonstrated the efficacy and preliminary safety data and toxicology data in preclinical study, we move into clinic, so into treating patients. And this is really what is the clinical, the trial phases. The trial phases, however, do not happen in one go, but in step. And that is what say, that is the trial phases, and these trial phases, phase 1, phase 2, phase 3, and phase 4. It's much more complicated, but I think that keeping this very simple structure approach help us to understand exactly what needs to be demonstrated in order to move to the next step. And I just briefly go through each of them. So the phase 1, I cannot emphasize enough, is a safety study. So when you take a drug from the lab to the patient, to participants, so to human being, the first thing that you need to demonstrate is the drug is safe. So the main objective of any phase 1 study is to determine whether the drug is safe. And in order to do that, you need to determine what is the maximum tolerated doses. And this is something that a lot of people talk about in clinical trial is the MTD. So the higher doses that can be administered without inducing side effects or severe side effects. Is what is the optimal biological dose, which in a way is the minimum dose to obtain the efficacy that you want to obtain. And these two values, so the maximum tolerated dose and the optimal biological doses, are the one that you recommend for the next steps. But phase 1 study are usually relatively small, can be, depending on the mechanism of action of the drug, can be conducted in healthy volunteers, but are usually done in a small group to determine this value. But you want to determine which are the doses that you want to take to patients and you want to take to larger cohorts. And animal models can support, but cannot answer this question. You want really to understand what is the dose that starts to show the toxicity, so you know that that is the maximum. And if you do not see any effect with the maximum tolerated dose, you cannot go higher. Is it pointless to have large study when you do not know whether the lack of efficacy of a drug is due to the fact that you have not selected the right dose, or is because the drug doesn't work? In order to do that, a phase 1 study first need to tell you what is the maximum dose that you can safely prescribe. The phase 1 study is when you want to study how the medicine go into the body. So the pharmacokinetics and pharmacodynamics. The pharmacokinetics is described as what the body does to the drug. So in general is how much of the drug the body absorbs and how long does the drug stay into the body. The pharmacodynamics is more are biomarkers of drug effect. You can have in phase 1 study some initial assessment of treatment efficacy. So you might want to see whether the drug does what it's supposed to do. However, if a phase 1 study design tells you that the main objective is to evaluate the efficacy, is either the wrong phase or the wrong outcome. But it's not something, first of all, you need to demonstrate to be safe before efficacy. Key elements for phase 1 trial, first of all, you want to choose a promising agent. So you need to have a strong preclinical data in order to move into a phase 1 study. If you have a weak preclinical data, you need to really go back before using humans. You want to define the population, but again, for the phase 1 clinical trial, you want probably not to waste patients that might actually be important to demonstrate a drug effect in later stages of the study. So usually phase 1 trials are really open to very general inclusion-exclusion criteria, but obviously you want to target the population that is the object of your study. As I mentioned, you need to decide which dose do you start. And usually depend a little bit whether it's a repurposing drug, so it's a drug that is already being used in human in another condition, and therefore you have more information about what is the dose that is prescribed in other condition. But if it's the first human trial, usually you go for the lower dose that you start is a tenth of the dose that is being used in mice, or a third of what has been used in dog. But this is really depending on the drug. So it's not a conclusion that you can make out for any approach. You want to decide how do I escalate? So how do I test higher doses? What is the process that I follow in order to guarantee safety, but to define the limited toxicity dose? As I mentioned, usually it's done in healthy volunteers, although in some, for specific mechanism of action, for example, for gene therapy, you cannot test in a healthy volunteer. So even phase 1 study are done in patient. So it depends. There are different models that can be done, can be single ascending doses model or multiple ascending doses model. But what you want is that you test escalating doses to reach the maximum tolerated dose. One of the models that is more follow-up is the 3+3 for phase 1 study, which means that you recruit 3 patients in at one dose. If everybody tolerates the drug well, you proceed with the next dose into the higher doses that you have defined as your next target. If you have one episode of one adverse event that is related with the study drug or that is considered a toxic effect of the drug, you recruit 3 additional patients on the same dose before escalating. If you have 2 or 3, so more than 2 toxic responses, then you stop and you decide that this one is the maximum tolerated dose. If you recruit other 3, then you apply the same. So, but if is a second step in a way, you have already shown 1 toxicity, you recruited 3 additional patients, you have 1 additional toxicity, you can proceed. But if you have more than 1, usually you stop, and that is the define. This is just one design. It's just as an example. It's probably the one that has been more commonly used, but in rare diseases, there are different models that has been developed and different study design, and all of them are valid as soon as there is the rationale behind the chosen doses, and as soon as safety of the patient is guaranteed, and therefore you always have a go, no-go approach. But because it's the phase 1 are a safety study, the primary endpoint is safety, so it's management of side effects, either clinically or biological side effects. And as I mentioned, always have pharmacodynamics and pharmacokinetics. Phase 2 study is when you, in a way, is the next step. And is actually you want to decide exactly the dose that you want to take for a larger study, and you want to confirm the safety of the drug in a larger population. But the phase 2 is when you start to introduce the concept of efficacy. So the phase 2 study wants at least to show that the drug does what it's supposed to do. A phase 2 study is usually not designed in order to statistically demonstrate the effect of the drug, but they need to demonstrate the biological effect of the drug as a minimum. And the importance of the phase 2 is the fact that phase 1 study usually recruit a relatively small group of patients, phase 2 a higher number, and phase 3 much higher number. And phase 3 study are quite costly. So the phase 2 is really to support which drug are more likely to success and therefore should we bring forward, and which one are actually not doing what we were supposed to do or what we serve in animal models, and therefore it's better to stop, think, and start again. We have to consider that approximately 20% of the phase 2 trial proceed to a phase 3. And as I mentioned, a phase 2 study, the main aim is to identify the correct dose, is to see whether the drug does what it's supposed to do, and to collect longer and larger safety data. Usually they are done in the targeted population. So you recruit a patient in a phase 2 study with a disease that is the object of the drug, because you want to measure efficacy. However, the duration can be really variable between 6 and 24 months, rarely longer, and rarely shorter, because in general, for rare diseases, it's very difficult to see a drug effect unless you have a clear mechanism of action in a period of time shorter than 6 months. They can be single arm if you have already identified one dose through the phase 1 study, or can be multiple arms and can be multiple doses, but can also include a placebo comparator. However, typically in a phase 2 study, the comparator is an external cohort or historical data. The problem with the phase 2, I don't want to go into many details, but quite often you can read this is a phase 2A or a phase 2B. If I want to summarize, the phase 2A is more a proof of concept, and the phase 2B is more the dose finding and efficacy study. So the phase 2A is what is really a phase 2. The phase 2B is more close to the phase 3 studies. And we will explain a little bit the phase 3 study. But how do you make the decision between is this drug working and therefore shall I move to a phase 3 study? Ideally, you want to show that your drug or the drug make a difference on something that is meaningful for the patient. However, what is meaningful to the patient quite often are hard endpoints. So you want to show that your drug prolong the survival, improve the quality of life, or if you have a clear disease event, in Duchenne muscular dystrophy, for example, is the loss of ambulation or the requirement of ventilatory support to breathe. So you have clear endpoint. If I can delay this clear clinical endpoint, you have shown that your drug works. The problem with the hard endpoint is that they quite often require very long interventions in terms of time. And when you need, so if you think about even Duchenne, that is a relatively progressive disease with children who die in their late 20s, is still a very difficult achievement to show that a drug improves survival, or you need very long trial, which are often not feasible. And then is when you use biochemical or biomarker endpoints, so something that tells you the effect of the drug, or surrogates endpoint, or that are other clinical factors that can predict the loss of ambulation, for example, or predict the survival. Phase 3 studies are usually larger studies, are the study that want to show that statistically significant evidence of efficacy and still collect the safety data. So usually are what we say is the pre-marketing study. So after that, the company can apply to ask for the drug to be approved and to be introduced in the clinical care of patient. So usually they select the dose, but what is important, the phase 3 study, in order to obtain regulatory approval, they need to show what is described as a non-inferiority model. So the results need to be compared with the gold standard treatment. So if you have a treatment available, whatever new drug that is approved as part of the standard of care need to be proven that is better compared to what is already available, or is not actually better, but is not inferior to what is available. Different way to do clinical phase 3 study usually are randomized placebo-controlled study, and I will explain why there is quite often the placebo, or in double blind in order to avoid any bias for that perspective. you usually are designed for specific population that are more likely to show the drug effect in the period of time that corresponds to the duration of the trial. So for example, for some condition, you don't want very severely affected patient, you don't want asymptomatic patient, but you want someone who you expect to deteriorate over the period of time of the trial. And therefore, you just need to show that the drug improve, slow down the deterioration, or stabilize the condition. So you could have just simply a low dose, high dose, and placebo group design, or you can have a combination when a low dose move then to a high dose, the high dose stay on the high dose, and the placebo go into a high dose. So everyone does a low, high dose and placebo phase, following by everyone receiving a high dose. Or you can combine in order to, for example, assess the effect of a delayed treatment. So, but this list could be much longer. I talk about phase 4 study because are part in a way of the didactical approach on clinical trial. However, recently in rare diseases, phase 4 study are becoming less and less common. And the objective of as a phase 4 study is to monitor the drug effect in the general population. So it's usually a post-marketing. The drug is already available on market, and you want to show the long-term effect of the medication. Are usually done on large population and on for much longer period of time. And this means that they are very costly, and that is why are usually now replaced with a more real data, real life data collection. We talk about efficacy. So the aim of the study of a clinical trial is to show that a new intervention is efficacious, so improved a clinical outcome. So there was a long discussion within the European Medicine Agency about endpoint. And I think that one important, for example, for Duchenne muscular dystrophy is that the regulator give you an idea, and there are guidance for outcome measure in several rare diseases produced by the EMA, and tell you a little bit what do you want to target. For example, for Duchenne muscular dystrophy, the main endpoint is to improve muscle strength and function. But clinically, we always thought that muscle function, so what the patient can do, is much more important. And the EMA, for example, agreed with that. However, they always said that if you can demonstrate that the patient improved in their motor performance because the muscle strength has improved, you have a stronger case. And therefore, they tend to recommend the collection also of a strength measure. And in general, the regulators, specifically the EMA, the FDA has a slightly different approach, but the EMA in Europe, they really emphasize like that whatever your outcome is should have a clear direct impact on the patient daily living. So if a drug is effective in doing something that the patient is not particularly bothered at, should that drug be approved or not? Again, the other recommendation in general is that you always need to choose one outcome measure that is the most important when you want to show the drug effect. However, the regulator recognized that what is actually important is more even of the statistical significance is the fact that you use different outcome, and different outcome go in the same direction. But as I mentioned before, there are some difficulties in terms of choosing the right endpoint. And this is when we talk about extrapolation and surrogate. For extrapolation, I mean, when you test, as mentioned, in phase 3, you test the drug in a population. That is the one that you are more likely to show a drug effect. But how can you then demonstrate that the drug works also in younger patients or in older patients? And this is an issue specifically for the European agency. And the extrapolation of efficacy need to be really developed based on real life data that allow you to say a drug works in this group and this group is different because of A, B, and C. And this is the effect that I see. A different component that is what, again, I mentioned before is the surrogate. So the surrogate is more than if I cannot have wait until to see if a drug postpone, for example, the loss of ambulation, which is not expected for maybe 10 years for when I do the drug. Can I find either a functional outcome or a biomarkers that correlate with that loss? So, for example, you can say that a specific mutation, for example, is associated with a you can say that, for example, the CK is not a great example, but it's probably a blood marker is very high, and you want to measure the drug effect if long term, and you want to measure whether this blood marker is reduced by the use of the drug. In order to be approved and considered by the regulator, you need to already show that if you have lower level of that serum biomarkers, you are clinically better. But if you have strong evidence that the biomarkers or that blood test result correlate with the function of the patient, that you are much more likely that the EMEA will consider that biomarker as a valid surrogate endpoint. The problem is that the surrogate endpoint need to be very carefully evaluated, because not all of them are good surrogate endpoint. Regulation also required for most of the clinical trial to have a placebo group. And why is so important? Is because, as I mentioned, if there is differences in how you treat the patient in your clinical practice, this might affect the evaluation of the drug effect. Although for rare diseases, the regulator also recognized the importance of natural history cohort and real life data to support, for example, clinical research in ultra rare diseases where a placebo control study will not be feasible because of the limited number of patients. So continuing that, the other aspect that I wanted to mention is some ethical concern about clinical trial in rare diseases. And I think that one of the critical point is to always recognize the concept of the clinical trial. A clinical trial is designed to demonstrate the effect, to evaluate the whether a drug is efficacious and is safe. And therefore, that is not an approved treatment, is not a treatment that will necessarily be associated with a clinical benefit. The clinical trial is really designed to answer this question. And this is important in all the process that all people involved, from the patient to the clinician, as this concept very, very clear. And then there are a lot of concern when I was saying that when you have a clinical trial, you need to evaluate against existing treatment. And how do you compare? How do you make the superiority model really work? Few considerations in terms of the ethical issues regarding then related to the consent. I think is absolutely critical. The consent process is a critical part of clinical trial, and you can't spend too much time to consent a patient. This is what, in a way, the consent allows you to go around several ethical issues because informed decision. So the part of the consent, the purpose of the trial must be clarified. You need to understand, to describe the trial design, whether there is, for example, a random assignment for treatment or placebo or different doses. You need to describe what are the trial procedure, how long it will take for each procedure to be conducted, how long the patient will be required to be involved in the study and to be involved in each visit. Taking part in a clinical trial might impact their daily life, and they need to be aware before making the decision to take part. They have to have absolutely clear in their mind, and you need to contribute to this explanation to inform them about what I was saying. That is not an approved treatment, it's not a treatment that has, for the benefit, has already been demonstrated in the majority of the cases. So what are the expected benefit? What are the foreseeable risk? But also make it clear that in the majority of clinical trial, the drug is a new drug and the safety profile in the clinical trial will contribute to develop the safety profile. So you contribute to write the leaflet that then you have with any drug that you buy in your pharmacy. So there are some side effects that might not be expected, but a safety monitor is in place for this purpose. You always need to remind that the participation is voluntary, is not a life-changing treatment. Therefore, the patient have always have the freedom to decide whether to participate in a clinical trial or not. And for this reason, they will not be judged or they will not be penalized in terms of care provision. You need to discuss the confidentiality of the data, who will have access to the data, how this will be shared, for example, with the sponsor, for the regulator, who will be able to see their medical notes outside their clinical team. It's important to say what is the plan in terms of sharing new information. And this means that, for example, if there is any safety concern, the sponsor has the obligation to inform the clinician, and the clinician has the obligation to inform the subject. And when relevant, the subject can change their mind because there is a new side effect. They need to have the freedom to decide to discontinue the clinical trial. But also in terms of what is how the end of the study result will be communicated. Each consent form needs to be able to provide a contact number that the person can get in touch if there is any issue or they have any specific question. And that is critical for the clinician taking part or leading a clinical trial, because he needs to be available or need to have a system in place for which a medical is reachable 24 hours. They need to understand the reason why a clinical trial can be stopped. And this can be something that they, regardless of the fact that they might see a personal or individual benefit, the studies can be terminated for lack of efficacy or for safety reasons. Patients cannot be compensated in Europe. They cannot be compensated in order to take part in clinical trial, but they can be reimbursed for anticipated and reasonable expenses. But other ethical concerns that are difficult sometimes for people to realize until you take part in a clinical trial yourself in a way, is the burden of taking part in a clinical trial. It's not like, in the majority of the cases, it's not like taking part in a routine clinical assessment. There are established visits that need to be done within a certain window, therefore the flexibility might not be as in clinical practice. The appointments usually are much longer, are much more frequent. They might need to undergo invasive procedures that are done only for research purposes. And one of the ethical issues, for example, is it ethical to expose a patient to an additional risk, for example, an anesthetic risk for research purposes? Where is the balance? And there is always a compromise for this reason. In order to try to reduce the burden to patients, you always need to compromise how many questions do you want your study to address, or do you want the study to address, and what is reasonable to be delivered within a clinical trial. And this is just an example to say how a clinical trial is designed. So this is the frequency of the visit, how many assessments, and from my experience, mainly in neuromuscular diseases, a clinical trial visit can last up to 6 hours, which is exhausting, and the patient needs to take into consideration the travel time in addition to the time that they will spend. They might get tired, they might get bored, but also they need to take themselves or the parents, if for pediatric study, they need to take time off in their daily life in order to complete the study visit. But a massive issue regarding the drug development in rare diseases is the drug cost. I don't know how many of you are aware. We have a few drugs, for example, approved for spinal muscular atrophy or Duchenne muscular dystrophy, who have raised these issues about how much does a disease, and it's very sad in a way putting in this term, but it's how much is worth it. So from the patient perspective, anything that improves their mobility or their quality of life or their condition is obviously worth it. But for the payer, which for example in the UK is the NHS, but is any national health system, need to counterpart to say how much is it worth, how much the national health system will save in terms of money if they decide to invest in this drug. And obviously, when we talk about a relatively small amount of money is different than when we talk about drugs that cost up to $300,000 per year, or even $1 million per year. And these are for individual patients. And I think that there are, I don't want to justify the drug cost in any way, and it's not my role and it's not what I'm good at, but there are some considerations that are realistic. So first of all, there are, from one perspective, the regulator has tried to incentive the development of a drug in rare diseases. For the company itself, for several years, there was the problem that investing in rare diseases for new drug wasn't very convenient, because if I find an effective treatment for, I don't know, for hypertension, I can target millions of patients and therefore have a lot of profit. For rare diseases, we are talking about a small number of patients, and therefore the market was not. And therefore the regulator, and this applies both to the EMA and the FDA, has developed what is called an orphan drug legislation in order to incentive company and have some economical benefit in investing in drug development in rare diseases. And there is also the fact that they might be able to have innovative trial design, because it's much more different to develop a clinical trial in hypertension compared to rare diseases. But also, on the other hand, the company itself, they said that the cost of clinical trial is very, very high. They have all the preclinical, and then phase 1, phase 2, phase 3, and potentially phase 4 clinical trial. And when they are in clinical trial, although they do not pay patients to take part, they need to reimburse the hospital for the service that they provide. They need to reimburse individual assessment. And these add to the final cost. But it's an issue, and will become an issue, I think, a bigger and bigger issues in several rare neuromuscular diseases, when now there is a lot of interest in terms of, for example, gene therapy approaches or gene modifier approaches that are usually quite costly. And how can the national health system sustain this long term? I just know that I tend to talk a lot, but I just wanted to spend the last few slides talking about why clinical trials fail. And what are some considerations that need to be taken into account. And this is really coming from the Duchenne community. Over the last probably 5 years, I've seen several clinical trials fail. But I would like always everybody talk about clinical trial failure. And I think that one important point that I want to mention is different to talk about clinical trial failing and drug failing. So the clinical trial, and I hope that is a clear message for today, aim to demonstrate whether a drug is safe and effective in treating a specific disease. But this is a question. So if at the end of the study showed that the drug is not effective or is not safe, that is a failure of the drug. The drug does not do what we were expecting the drug to do. But the study, the clinical trial has not failed. The clinical trial has answered the question that was supposed to answer. A clinical trial fails when it does not reach the conclusion of the answer. So a clinical trial fails because at the end of the study, we do not know whether the drug is safe or is effective. And this can be due to the fact that it wasn't designed properly, wasn't powered properly, so they did not recruit enough patients in order to make this conclusion, or they struggle with recruitment or run out of money. This is a clinical trial failure. The other are drug failures. And in Duchenne, we had several drug failures, not clinical trials. But why do they fail so much? Why don't we have an effective drug in Duchenne? So one consideration is, for example, is that you need to take into account is the complexity of the disease. When we talk about rare diseases, think about what is the organ that we target. For Duchenne muscular dystrophy, for example, we target the skeletal muscle. The skeletal muscle is a huge component of our body, so represented up to 30 to 40% of the full body mass. And therefore, the tissue that you target is quite extensive. How can I reach with an intervention such a large tissue? Is the fact that maybe it's not just one target. You might not target only the skeletal muscle. For example, in Duchenne, also the heart is affected, the breathing muscles are affected. How can I target all these different organs? And what is the cause of the disease? It's genetic. Modify the genetic cause is not a straightforward message. So it's just to find a way to treat a complex disease is complex itself, and is a good reason why it's difficult to have a successful drug. But also, as several rare diseases, if the disease is progressive, such as Duchenne muscular dystrophy, the majority, is a cascade. Once you have something that has been lost, and it's very difficult that any drug will replace or will retrospectively so correct what has been already lost. So what is the best time of intervention? If you do not target the pathomechanism, or you don't target all cells that are affected, you have anyway the cascade process. And depending on how much of the tissues has been targeted, you will have an effect, or the cascade process will be more important. And we have example in Duchenne muscular dystrophy or study that has failed. And I don't want to go into the details of this clinical trial, but what I wanted to mention is the first large clinical trial in Duchenne who failed to reach the statistically significant on one outcome measure, that was the 6 minutes walking test. And one of the problems is that they saw that they tested the high dose, the low dose, and then the placebo. The low dose was doing possibly a little bit better, but the high dose was exactly the same of the placebo. However, if you look at this schematic, there is a concern to the fact that in a way, the only statistical significance difference of the low dose of the drug was seen at 48 weeks. Otherwise, they were running parallel and was not statistically significant. But this study, go quickly here, but this study used an outcome measure that was the 6 minutes walking test, for which we knew very little about in Duchenne muscular dystrophy. And therefore, only after the study, we discovered that actually the natural course of these outcome measure without intervention means that some patients tend to be very stable and some patients need to progress very rapidly. If you do not know your outcome measure, it's very, very difficult to measure a drug effect. So you know, there are, for example, in Duchenne, several other outcome measures, but you really need to know your outcome measure in order to be sure how does it behave in a population that has not been treated with a specific intervention, and what is the expected effect of this intervention. There's been a lot of work in terms of how do you define outcome measure in, for example, in neuromuscular diseases, and there's been a full workshop and a lot of work in terms of international collaboration and work with the EMA. And I think that the general message is really, you need to know the natural history of your outcome measure. There isn't really one ideal outcome measure, but the outcome measure depends also on the disease and on the disease progression and on the disease phase. And more importantly, is that you might need to have several outcome measures that go in the same direction in order to demonstrate a stronger drug effect, instead of having only one specific outcome. Knowing which factor can influence the outcome measure and the natural history of that outcome measure is absolutely critical. More importantly, you need to have strong preclinical data. So I can't emphasize enough that at the end of the day, a drug doesn't work if a drug fails. So we do not have a drug approved because we do not have good drugs. And where you learn about a drug is really from the preclinical. And is it critical, for example, that to verify whether preclinical data come from more than one lab and more than one resource. So I hope really that I convince you that strong preclinical data are critical, that is important to really well design your study in terms of choosing the right primary and secondary outcome, the duration and the population that you want to target. And the drug failure and the trial failure are two different concepts or two different aspects. Thank you.
Thank you, Michela. Brilliant presentation, a real manual on how to go through the process. So I enjoyed it very much and hopefully everybody will. Usually we have questions. Today's presentation was a bit long and our audience has not put any questions, but I'm going to put one if you allow me. So it's very interesting what you said about treatments and about the framework where new treatments appear, and you talked about several treatments, and in fact, the treatments for SMA that you mentioned are a case of success. At present, they are a very good propaganda for continuing to the effort to development treatments for these rare neuromuscular diseases. You talked about valboron, and you talked very well about how do you consider that a drug is not only efficacious, but it's worth paying for it. And you mentioned that many times what we do require is non-inferiority of the drug when compared with the standard. And usually we are happy that the drug has some less side effects than the standard treatment. And that's probably what's going to be demonstrated about valboron, at least initially. But namely, when you consider, as you said, that drugs nowadays have prices that are a bit over the moon, so when should we start thinking about demanding superiority studies instead of non-inferiority studies?
That is a tricky. I mean, if you think about the problem in a way with vamorolone, for example, is that although corticosteroids are routinely prescribed, there is only one licensed steroid in Duchenne muscular dystrophy, that is the Frasacor, which is licensed only in the US. So we don't have actually a licensed drug in Europe for Duchenne muscular dystrophy, and therefore it's very difficult to say which is the superiority against what. At the same time, if we look into that, it's not as straightforward because the regulator has asked a so is still a non-inferiority. They actually still ask for a superiority component. So for any drug, you still need to demonstrate, if there is an approved drug, you still need to demonstrate that it's better compared to what is currently available. The point is that the superiority can be either in terms of efficacy or in terms of the side effects profile, not necessarily for both of them. This is valid for licensed drug, but for example, for Duchenne, vamorolone will need to demonstrate that it is superior to corticosteroid, and what the company has chosen that is to demonstrate the superiority of the drug versus corticosteroid in terms of side effects, but not in terms of efficacy.
Okay. Now we have several questions from Onur Kakir. You can unmute yourself and voice them. Otherwise, I will read them, but perhaps it would be more interesting. Okay. In that case, hello? Yes, I'm going to read them.
Okay, I think I have managed. I'm outside, so I just wanted to be silent if you know, it's
okay.
not clear what I say. Yeah, first of all, I'd like to thank for this presentation. I really appreciate. I'm from Turkey, Istanbul, Turkey. I have a daughter with a with a disorder related to collagen 6 mutation. So my questions are related to that possible clinical trial that we have been expecting. So I might have missed the answers in your presentation. So sorry for that, but I wanted to get your answers again for these three questions.
So the questions are the
let me. I lost. Okay, now I see. Yeah, the first one is the main general prerequisites for the clinical trials for neuromuscular diseases. Just to trying to understand who are selected. Is there any general prerequisite for the clinical trials of neuromuscular diseases, or does it vary from disease to disease? My second question is there a specific time period? How long do the clinical trials continue for again, specifically for the neuromuscular diseases? And my last question is, what is the percentage of the clinical trials which haven't proceeded to the final drug approval in neuromuscular disease? Hope everything is clear.
Yeah.
Thank you
for your questions, and just try to answer as much as possible. So
Thank you.
in terms of the prerequisite for clinical trial neuromuscular diseases, who are selected is really depend of several factors. It depend on the disease itself and depend on the drug itself. So for that perspective, there are different components that to say. So which disease is picked? So why do we have so many clinical trials in Duchenne muscular dystrophy and in SMA and not so many in other neuromuscular diseases? And one of the reasons is also is related to how many patients. So Duchenne muscular dystrophy is the most common and is most severe muscular dystrophy in pediatrics, and therefore is why it's probably one of the targeted when other neuromuscular diseases, including collagen 6 diseases in pediatric, are much rarer, and therefore clinical trials are very difficult. But in terms of the population that is selected within, is really depend on the disease, on the mechanism of action of the disease, and the mechanism of action of the drug that you are tested. In general, for neuromuscular diseases, we need to consider that once they are progressive condition, and usually are associated with the fact that you lose muscle. So when the muscle is lost, it's very difficult to have a drug that reverses the process and give the muscle back. So in general, the population that are targeted are younger population where the muscle is better preserved to expect a more long-term effect. But this doesn't apply 100%, depend again on the drug itself. And there is becoming more and more clear that different population might provide different information on the effect of different drugs. How long the clinical trial continue? Again, I can simply answer it in a way to say that depend on the drug and depend on the disease. But the reality is actually there is another significant component about that, is that how much the company want to invest and how much want to invest in the disease itself in a way, but not necessarily in a negative perspective. So if you ask the company, they want to have a clinical trial that lasts 3 months and give you the answer. But then we know that this is not feasible in diseases such as neuromuscular diseases, where the progression is much slower, and in 3 months you will never have this answer, whatever drug you use. If you ask a clinician, I would say, I want 10 years, so I want to see whether really the drug prolong the survival. But that means that the drug will not be approved probably for 30 years, so convenient. So it is something in between. The majority, and depend also on the phases. So phase 1 clinical trial are much shorter than phase 3. For a phase 3, that is a pre-marketing, the duration usually is a minimum of 1 year, and usually up to 3 years. What is the percent of clinical trial which haven't proceeded to the final drug approval in neuromuscular diseases? I have this graph, and then actually there are different stages of failure for that perspective. Is how many drugs do not go, do not move from preclinical to clinical, and that is where we lose probably the majority, with no doubt, up to 80%. Then you have how many phase 1 study do not move to phase 2, how many phase 2 study do not move to phase 3, and how many phase 3 do not actually reach approval. For Duchenne muscular dystrophy, we need to consider that we have only a few antisense oligonucleotides that arrive to drug approval, and only, for example, the antisense oligonucleotide in US, Translarna in Europe, so not consistency across the globe. And currently in neuromuscular diseases, we really have a very limited number of drugs that has been approved. So the large majority do not reach that point.
Thank you very much. I appreciate it.
You're on mute, Antonio. Antonio, you are on mute.
Sorry. So I was saying that thank you very much for your presentation, Michela. We are a bit advanced in time, and so we have to wrap up. And it was very instructive, both to the ones that attended live today and to the ones that will see the webinar as a recording through the ERN website. Thank you very much, and talk soon, I hope. Bye now.
Thank you.